Good morning, ladies and gentlemen. As we have gone through this unit so far, you have become more acquainted with Punnett squares, monohybrid crosses, and dihybrid crosses, and I put together the screencast for all of you to hopefully be able to understand a little bit better exactly how to use these and what they mean. This here is an example of a Punnett square that you will typically find. Here it is. This particular Punnett square is demonstrating what it would look like if an organism were to breed with another organism of the same species, and we're testing for the probability of one particular trait. So on the top here, this blue arrow is indicating one of the parent. This particular parent is homozygous recessive for the color allele. In this particular example, that means that this parent is green. This blue arrow over here on the left is signifying a parent that is heterozygous dominant. Or you could just say heterozygous, meaning that this particular organism has one dominant allele and one recessive allele, making this organism yellow. Now when you complete a Punnett square, there's nothing really too special about it or anything too difficult. It's basic math, and it's also a little bit fun to kind of see what will come out. So what you do is you take the top parent, the homozygous recessive green plant, you take the one on the left, the heterozygous yellow one, and you ultimately combine them. So what you do is you take one allele for the particular genotype, and you put it on each square of the Punnett square. So you're going to have a big Y, a little y for this parent. For this parent, you'll have two little y's up here. And then ultimately what you do is you combine the two alleles. So here you have the yellow Y, the big Y, moving to here, and the little y from this parent moving to here. That gives you the heterozygous genotype that you're seeing over here. It's the same thing on this far right corner, this upper right corner. You take the big Y, you take the little y, you get the same result. On the bottom here, it's a little bit different. You do the same principle, but you have a little y indicating green from the parent on the left, and the parent on top you have a little y indicating green, giving you a y result. And it's the same on the lower right-hand corner as well. So now that we're able to fill out this Punnett square and kind of understand exactly how these alleles add up to make these genotypes, now we are able to look at what this means to us and predict the possibility of a particular trait arising in an organism. For example, this heterozygous uppercase Y and lowercase y indicates yellow, as you can see, the parent on the left. If there is a dominant allele in an organism, which is the uppercase Y, the organism will express that trait. However, in these two bottom genotypes, there is no dominant allele, it's just two lowercase y's. That means that this organism will express the recessive trait, which in this case would be green. So if you were to calculate percentages, there are 1, 2, 3, 4 squares. Out of these 4 squares, 2 of them are heterozygous, meaning that they will express the yellow color. The other 2 are homozygous recessive, meaning that they express the same allele, the recessive one, and they will appear green. Meaning it's a 50-50 chance that this particular, if these two plants were to breed, their offspring would appear green or yellow. The Punnett square side I just recently showed you is essentially a monohybrid cross. In this example, both organisms have the genotype uppercase B, lowercase b, making them heterozygous. And again, for clarification, a genotype is just two alleles mixed together. In this particular case, it's just two alleles, the capital B and the lowercase b. Typically, as I mentioned earlier, capital letters are used to indicate dominant alleles, and lowercase letters indicate recessive alleles. So if you're to see the genotype BB, capital B, lowercase b, that means that this organism will express whichever the capital B indicates. When you mix two heterozygous organisms, as you see here, you're going to have a constant ratio of genotypes. That's expressed over here. You're going to have 1/4 of the organisms be homozygous dominant, which means they will carry and they will express the dominant trait. Half of the organisms will be heterozygous, meaning they will express this trait, and the remaining quarter will be homozygous recessive, meaning they will not express this trait, if that is the case. On the left here you see an example. It's very similar to that slide I just showed you, the Punnett square. It's the same process. You just combine the two alleles and you get these as a result. And these results over here match up with the percentages I have indicated by this green arrow. Now, although these particular ratios up here demonstrate what the possible genotypes could be, they don't necessarily let you know what the phenotype or the appearance of the organism will be. Down here in this little passage I have underlined, 75% of the offspring will be black, meaning they have to have the dominant allele. It could be homozygous dominant, two capital B's, or heterozygous. So you have 1, 2, 3 of the 4 have that capital B, 75%. While only 25% will be white. And since white is the recessive trait in this particular case, they cannot have a capital letter, which indicates a dominant allele, and only one of these does not have a capital letter, again, 25%. So as I outlined down here, the ratio of phenotypes is 3.1 for this example, or 3 to 1, which is typical for a monohybrid cross. Moving on from a monohybrid cross is the dihybrid cross. This is a little bit more complicated process, but if you pay attention closely, it'll be easy to grasp and you'll be able to understand and apply it. So the difference between the two is a dihybrid cross looks at 2 or more genes in an organism, where the monohybrid cross and your typical Punnett square only looks at one. We were looking at black. We were looking at green versus yellow pea plants, for example. Also, the Punnett square only works if the genes are independent of each other. So the Punnett square is nice, it's easy, but it does have its disadvantages. And this is where the dihybrid cross comes in. So for example, you see this much more complicated little Punnett square down here. This is illustrating a dihybrid cross between two heterozygous pea plants. Again, hetero, different, so that means they, for these particular genotypes, these organisms do not express two capital letters or two lowercase. And in continuation with that, the genotypes are actually capital R, lowercase r, capital A, lowercase a, heterozygous. So if you have that, if this plant were to breed with itself, for example, these are the possible genotypes here. Because you have a capital R, and that could be taken with a capital A. A capital R could mix with a lowercase a, and so on and so forth. So you have two plants that are expressing the exact same genotypes up here. So now we're breeding them to test for two different traits at once. So we'll say, for example, capital R stands for round and lowercase r stands for wrinkled, while capital A stands for green and lowercase a stands for yellow. So if capital R stands for round and capital A stands for green, this one is clearly round and green. This one is round and yellow. This one is wrinkled and green, and this one is wrinkled and yellow. It's the same over here. So there's nothing tricky about mixing these two, it's just the same, but mind you, you will have 4 letters in the body of your square here versus the 2 that you saw in your Punnett square. So you combine your 2 R's, your 2 A's, meaning this one is homozygous dominant for wrinkled and for green. Now typically after you complete this square, you will find that your results display a ratio of 9 to 3 to 3 to 1. That is typical for a dihybrid cross. It doesn't mean it'll always be like that, but it should be close. So if you're experiencing something different, you might want to take a second look at it and see exactly what's wrong and what needs to be fixed. If you're finding that the dihybrid cross, that 16 column square, is a little bit more difficult to complete than the Punnett square, you can still use the Punnett square to test for 2 different traits, and I'm about to show you how. You use something called the tree method. And what you do is you complete 2 little Punnett squares. You all should be masters at these by now, I would hope. Up here you have, again, your genotypes testing 2 different traits at once. So I understand the alleles change. You had an A in the last example, but that's not very relevant at the moment. You have round and yellow. Capital R obviously means round, and capital Y signifies yellow. So you do the same thing, except this time you're just testing for one particular trait. You're not going to have 4 letters in here. You will only have 2, like the first example I showed you. So each parent is heterozygous for both traits in this example. So you'll have capital R, lowercase r, capital R, lowercase r, and then the same down here with Y's. So after you are to complete your 2 Punnett squares, then you have a little bit of math involved over here. So for this Punnett square on the top, you choose one of the 2. We're going to choose the one on the top to begin with. We have 3 different possibilities. It could be homozygous dominant, but there's only one of those. So a fourth are 2 capital R's. There are 2 heterozygous genotypes, and out of 4 that gives you half. The remaining one is homozygous recessive, so you put that here. Then you're going to have to do a little bit of math, but it's not too difficult. Each of these 3 is going to have 3 branches, as you can see. This particular illustration does a very good job of showing you what this should look like. And all of these 9 possibilities here are going to be the same for each one of these. So what you do then is you look at the second Punnett square that you haven't used yet. That's the one on the bottom for us. And again, you look at the results. Out of the 4, only one of these is homozygous dominant. It gives you a fourth, 2 capital Y's. 2 of them are heterozygous. It gives you a half. The remaining one is homozygous recessive, so 1/4. And you do that for all 3 of these on the right here. After you do that, that's when the math comes into play. In order to find out the potential percentages of a trait arising in an organism, you simply multiply the 2 fractions and basically add the 2 genotypes. So for example, 1/4 times 1/4 is 1/16. You have 2 capital R's, 2 capital Y's. So 1/16 of the organisms that these 2 plants could potentially breed would be homozygous dominant for both round and yellow. One more example. You have 1/4 times 1/2 gives you an 1/8. You have 2 capital R's, a capital Y, and a lowercase y, meaning 1/8 of the organisms could be homozygous dominant for round and heterozygous for yellow. And you do that until you have all of your percentages figured out. But again, these are just percentages, or in this case fractions, of the genotypes. It doesn't necessarily tell you what the organism will look like. So in order to determine what an organism will look like, for example, this one will be round and yellow, this one will be round and green. You have to go through and determine which each of these genotypes would lead to And after you do that, you should again have a ratio of 9 to 3 to 3 to 1. Again, this is just another good example demonstrating what this should look like. On the right here, you just have a switch is essentially a Punnett square on steroids, if you will. Again, it's got 16 different squares versus the 4 you'll see, but that's because each parent has 4 separate genotypes, for these particular traits. And again, there's nothing too complicated about it. You just combine the two. Each parent brings a capital R, each one brings a capital Y, making this particular offspring homozygous dominant for each trait. Capital R means round, capital Y means yellow. This one will be round and yellow. You do that for all 16. And again, down here, you see that each one contributes to that 9 to 3 to 3 to 1 ratio that's common of a dihybrid cross. I like this particular example because it includes what the organism will look like. This is a round yellow, particular P. This is a wrinkled green. This is a wrinkled yellow. So really look at this if you are struggling. These numbers here kind of signify that each of these squares is 1/16. So when you count up how many round yellows you have, you're going to count 9 in this little row. That's 9/16. So that's essentially where you get that ratio from. Now, ladies and gentlemen, if you could please pull out your notebook, I would like to do an example of a Punnett square and a dihybrid cross with you to make sure you really are getting it, and I can address any questions if you still have them. So for this particular example, we are going to mix two parents who are breeding for the particular trait of hair color. And each parent brings a different genotype to their possible offspring. So the parent on top is going to be homozygous dominant for black. So what would that look like? Just think about it for a moment or two and write down on the top of your paper what it would look like. For me, I put 2 capital B's. It's homozygous dominant, meaning they will both be capital, and B for black. It's pretty simple. The other parent is going to be heterozygous for black. Again, B is for black. A lowercase b would signify white. Hetero means, class, different. Very good. So, if it's heterozygous, one has to be capital and one has to be lowercase, correct? Now, some students may think that, well, what's it matter if I put the lowercase b over here and the uppercase b? It won't affect the results, but I guess it looks nicer, and it's more common to put the dominant allele first, as you saw that I did. Now, as I showed you earlier, it's pretty simple. You just combine the two alleles to get your different genotypes. So in the upper left-hand corner, what will we have? Very good. We will have 2 capital B's. How about the upper right-hand corner? Again, it will be the same as the upper left. 2 capital B's. How about the lower hand left corner? What will we have? Very good. We will have heterozygous genotype, and the same for the bottom right. Now, here's where your two little ratios are different. If you look at the results we had in the Punnett square, each one has a capital B, correct? Yes. So if that genotype does have a capital B, that means it will express the dominant trait, in this case black hair. So that means 100% of organisms would have black hair if these two parents were to mate. However, that does not mean the genotypes are all 100% the same. Up here, as you can see, we have homozygous dominant, but down here we have heterozygous. So that means 50% of the offspring could be homozygous dominant. The other 50% will be heterozygous. And in appearance for the offspring of these two parents, it won't really play a large part because each one will have black hair. But for the offspring's potential offspring, or the F2 generation, if you will, it could come into effect there and play a larger role. Now we are going to do an example of a dihybrid cross. So underneath the area in your notebook where you just completed the Punnett square, please make a square with 16 blocks in it so we can complete this process. Now that you have done that, I will let you know that we will be having two parent organisms mating, and each one will be heterozygous for two traits. So what does that mean for the potential genotypes that we will be placing on the top and on the left of the square? So for example, one parent is heterozygous for round, as we saw in one of our previous examples, but please do not look back. That same parent is also heterozygous for the color yellow. So knowing what you now know, could someone please tell me a genotype that I should put on the top left? Very good. To begin with, you start with homozygous dominant for the two main traits, round and yellow. And as you move to the right, you become heterozygous. So that utilizes the two capital R's. And as you keep moving on, since the organism is heterozygous for round, that's where the lowercase r comes into play. It's also heterozygous for yellow, so the big Y comes back into play. And since I mentioned both parents were heterozygous for these two traits, what should it look like on the left-hand side? It should look exactly the same. So as I'm filling this in, some of you might feel overwhelmed at the moment. I want you to realize that it really is no different than a Punnett square. You just add an additional allele. So as we go through, it's what we've been doing the entire time. Now since I don't have a large amount of room in these particular squares, if I were to do it on the board I would make the squares larger obviously, I will be writing the two genotypes on top of each other. So don't get confused, pretend that they are combined. So for the first one on the upper left hand corner, what would this look like? If we were to mix these two alleles from each parent, or these two genotypes, sorry. Correct, we would have two capital R's, two capital Y's, because that's what each parent brings. How about the second to the left hand side on the top? Exactly. We would have, again, two capital R's, but this time capital Y and a lowercase y. So now that you see me do two, please go through and try to complete the rest. And in a moment or two, I will put this back up on the board, and you can see how you did. So after you took a few moments and were to complete the square, this is what it should look like. Now please bear with me. I understand Mr. Ames is an artist, and this is a little bit sloppy. If there is anything you're unclear about, you can ask me, and I can clear it up for you. But essentially, this is what yours should look like. Now again, if you were to go through and were to complete a ratio of the genotypes, it would look much similar to that tree method that I showed you that requires a decent amount of math. So for example, only one of these potential organisms is going to be homozygous dominant for both. If you look throughout, I don't see another one. Again, I only see one homozygous recessive for both. That's a genotype ratio. To complete the phenotype ratio, we should, again, experience a ratio similar to 9 to 3 to 3 to 1. And we would get that if we were to go through. This one on the top is round and yellow, correct? The one right below it is round and yellow. In fact, all of the organisms in the left-hand row are round and yellow because they carry at least one dominant allele for round and at least one dominant allele for yellow. I hope you all found this particular screencast at least somewhat helpful in terms of using a Punnett square and the difference between a monohybrid and dihybrid cross. I want you all to realize that these are not complicated processes. You should not be afraid of them. Although they may be new to you, with a little bit of work and determination you can get through it. And please know that I'm always here to answer questions and I'm more than happy to help you. But also know that your peers are very educated individuals and are more than willing to share that knowledge with you as well. So please do not hesitate to ask if you have any questions, and I hope you are enjoying this unit as much as I am. Thanks.